All right, guys. Today we're gonna you're gonna sit here and listen to me talk about DNA replication. Last time you watched a video of a guy who has been talking about or had been talking about DNA structure, which most of that stuff should be review for the most part. It's all pretty simple stuff. There were a few new ideas I think that were in there that I think we'll hit on again this time. Those are this idea of 5' and 3', and you'll see that pop up again in this lecture and realize why, pardon me, it might have been so important. So we're going to get into um, DNA replication. And the reason that we needed to know the structure, or that the structure is so important, because it suggests that there is a way that this new molecule that Watson and Crick had kind of discovered the structure for could actually replicate or make a copy of itself based on the structure that was there. They looked at it and said, wow, this looks just like something that could make a copy for itself. So it provided this mechanism for copying. So that's what we're going to get into today. Now, this is a process, DNA replication, that is known to be what is called semi-conservative. And I'm kind of going a little bit out of order, but I'm going to skip some of the slides coming up here shortly. What that means is that when you copy this, when it goes through synthesis, DNA synthesis and mitosis or meiosis, um, you are taking part of the original DNA strand and you are going to incorporate it into the new DNA strand. So what you're going to use is basically a template. So if we look at the sequence of pictures that we see right here, this is the parent molecule, parent DNA molecule that we see in front of us. Oftentimes our book, the animations, the stuff you'll see in my PowerPoints shows that these parent molecules are dark blue, and that's how you can tell what's the parent molecule. And then the light blue is oftentimes the daughter molecules, the ones that are brand new. So here we see the parent molecule. Both strands are dark blue. Each base is paired with its complementary base, and it has yet to do anything. It hasn't started DNA replication. In this next picture, we see that it is ripped apart. And basically what we have on each side, we have a template here, and we also have another template here. Okay, this is like a docking point for new base pairs that are going to come in. And basically what we're going to see as we work our way through is that these new base pairs during replication, since these two template strands, this one and this one, are separated from one another, we can actually add new bases to these open parent base strands right here. Okay, so we add new bases right here, which we see taking place right there, and we add new bases on this side as well. And so what that does is it provides two templates that are kind of frameworks for new bases to come in and make new DNA strands. And so ultimately what we get at the end is two DNA strands. There's one, and then here's the second one, that are identical to this parent strand. Okay, so we have replicated the DNA. Now, this term semi-conservative, these are the slides I'm going to skip right here. But this term semi-conservative, if you think about what semi means, it means half. Okay, half of this original DNA strand right here is conserved in this new DNA strand right here. Okay, the other half, this side right here, is conserved in this DNA strand over here. Okay, so that's why it's called semi-conservative replication, and that's really the most efficient, easiest way for DNA replication to happen, is to have this double-stranded parent that we see over here split apart, and once it splits apart, you can add new bases like we see here, and basically what you're getting is two identical copies. One of these will go in one cell, and another one will go in another cell. Okay, that's what happens in mitosis. Okay, go ahead and skip these. They're interesting. It discusses some of the different models and experiments that were done to kind of verify that semi-conservative replication was in fact the way that this actually occurs. There's different hypotheses as to what actually happened at one point. We're not going to go through that. It's not required that you know that. So, all right, we're going to get deeper into this this process. For the most part, it's a pretty straightforward process. There are some hiccups to be talked about a little bit more in detail, and we can go through those if you need help. I've got a resource that I put together that will really help you if you are having difficulties with some of it. But anyway, let's talk about how this starts and then where it goes from there. Replication starts at multiple places, multiple um, points along the genome, okay, on every chromosome. And those points of replication starting are called origins of replication. Okay, they're just little points, little base pair segments where what happens is the DNA starts to open up, or the double strands start to separate from one another, and it causes what's known as a replication bubble. And when we see a replication bubble, ultimately what we get is replication proceeding in two directions. So we can imagine, and we have to kind of use our imagination here, but at this black point, right where the laser pointer's over, and at this black point down here, those are the origins of replication. At some point, they were closer together, they were connected. So the dark blue, again, here's our parent strand of DNA, and here's the other parent strand of DNA. They were connected, right? And during the starting points of replication, this origin of replication is where they started to separate from one another. And so that's what we see right here. Now, this, like I said, this happens at multiple points along the DNA strand, along the chromosome. If it only happened at one point, it would take forever for replication to occur. It would not be efficient enough for us to make copies of ourselves in a meaningful time. Okay? So once we get these replication bubbles open up at the origin of replication, these pink arrows indicate the direction of replication. And so what we see, if the origin of replication started right here where the red laser pointer's at, we notice that replication occurs to the left, pardon me, and also to the right of that point. Okay, and so the bubble gets bigger as we see. Ultimately, these bubbles, these replication bubbles, will merge when they meet another replication bubble. Okay. Now, this term right here, the replication fork, that is the point at which the original parent DNA strands have yet to really separate. It's the point at which they're going to separate, and that's important. It's a nice landmark because it tells us what's coming next. It allows us to understand where things are going and where they've been. So the replication fork is a term that you will hear me say again as well. So that's in general, that's how DNA replication starts, and we're going to get even more detailed as we go through it. Now, to add a little bit more detail, what we're doing again, once we separate these parent strands of DNA, these dark blue ones that we see here, what we ultimately want to do is lay down new base pairs, new railroad track, if you will. I call it that a lot. And it doesn't just happen by magic. There is something that helps helps these light blue base pairs be laid down, and that something is an enzyme called DNA polymerase, and we know it's an enzyme because it ends in ASE. So this enzyme called DNA polymerase actually catalyzes the attachment of a new nucleotide to the growing new strand of DNA. Okay. And so, when we see it in animations, and I'll link eventually some animations to this. I'm not going to do them in here because it takes too long for the thing to upload. But when we ultimately see the DNA polymerase add these things, it looks like this big blobby thing. And what that big blobby thing is an enzyme that catalyzes or facilitates the attachment of the new nucleotide to the growing strand of DNA. Okay. And here's just some kind of fun stats. It goes quick. Okay, the DNA polymerase can add bases in a matter of 50 per second in humans, and even faster in bacteria. It doesn't matter as much in bacteria because they can deal better with mutations. But with human cells, it goes a little bit slower, even though 50 nucleotides per second is extremely fast. The 500 nucleotides per second in bacteria is a lot faster because they don't care so much about mutations. In fact, sometimes they benefit from those. But just a couple of FYI things. But this is a diagram that shows us what's happening. when we add new bases. Okay, so this is the growing strand. It's going to grow one base pair bigger as compared to this one over here. So we compare. It's almost like they've got it set up almost like a chemical reaction with a little arrow in the center and the enzyme right over top, like we've seen before in some of the other chemical reactions, except this is a giant chemical. This is a really big molecule, right? And so that's really the only difference. But what we see here is we see the parent strand, again, in dark blue, okay, acting as a template, and we see the growing new daughter strand. This is the new strand over here. And what we see coming down right here, this guy right here is the new nucleotide. This is the new nucleotide that's going to be added to our growing DNA strand. Okay, it is yet to be added. And the one we're going to add here is T, thymine. And we see that it is not added here. And then over here, it has been added. Okay, now what's changed? The fact that we added DNA polymerase. Okay, so DNA polymerase facilitates the attachment of this guy right here to this hydroxyl group right here. Okay, that's now this is where we get into this idea of 3' and 5'. Okay, in the next slide you'll see here it talks more about elongation of that new strand. New base pairs, okay, like this T that we're going to add right here, can only be added to the growing 3' end of the new strand. Okay, so that's what we see happening here. This right here is the 3' end. So here's the 3' end of the growing strand. See how it's growing? It started small up here, laid down more and more and more. So this is the 3' end of the growing DNA strand. Okay, new base pairs can only be added to this 3' end because this phosphate here is going to basically attach to this OH group here. That is the chemical reaction that needs to happen for this base pair to be added. Okay, and DNA polymerase facilitates that. So another way that we could say this is that a new DNA strand grows in a 5' to 3' direction, meaning the new one started up here. This was the first base that was laid down, right there. This is the next base that was laid down. This is the third base that was laid down. This is going to be the fourth one. So it starts at the 5', the new strand right here, starts at the 5' end and grows in a 3' direction. Okay, so it goes from 5' to 3'. So that's why the discussion in the structure that maybe we hadn't talked about, you hadn't learned before. That's why this idea, this understanding of this 5' designation of this end of the DNA and the 3' designation of this end mattered so much, because you can only add these these new base pairs to the growing 3' end down here. So we see it, how it's been added. Okay. All right, and that's basically what this slide says here. Now, this happens in an anti-parallel way, meaning if we go back one slide, we would have other instances where I'll go to this one. This side, if we wanted to build one from this side, it would have to grow from this side here. Okay, so this one, if this was the new one, it would grow from the 5' end here all the way up here, which is anti-parallel or opposite the way this one's going from the 5' end here to the 3' end down here. Okay, so this is anti-parallel elongation. All right, now one thing we have to keep in mind here is that a new strand can only elongate towards the replication fork. Okay, if we go back a couple of pictures, you notice that these pink arrows, which indicate the direction of replication, they're only happening towards the replication fork in either direction, and that's going to be something that causes a little bit of problems when we get into discussing how the entire DNA strand, both double sides of it, are taken into account. Okay, So when we are creating the new DNA strands, when we're making two brand new strands, they happen in two different ways. Okay, one of them, it happens uh, by what's known as the leading edge. Okay, the other one is a lagging edge or a lagging strand. So um, when we are discussing these two different fashions of creating the DNA, we call one the leading strand and one the lagging strand. When we're talking about the leading strand, DNA polymerase can work continuously. It lays down base pairs without ever stopping. It's as if it starts once and just keeps going. Like it looks like a little typewriter. Now the lagging strand is where people start to get a little bit more confused about what's happening when the DNA base pairs get laid down. In the lagging strand, the DNA polymerase must work in a direction away from the replication fork, but the overall direction of replication still has to happen towards the replication fork. And so what this creates are these little fragments of DNA. Instead of one long continuous leading strand, we get these little chunks called Okazaki fragments. Ultimately, the Okazaki fragments are joined together by what's known as DNA ligase. I'll talk about that a little bit later. But the DNA ligase basically glues those little chunks together. So it's DNA building normally. It's not, maybe not normally, but it will yield a normal DNA strand. So it's nothing to really worry about. It just has to be done in a particular way because we know that DNA bases can only be laid down, pardon me, in a 3' manner from a 5' to 3' end. Okay, so this diagram here kind of gets us a little understanding about what's happening. Okay, so think about how this started at some point. This red dot up here in the upper right-hand corner maybe was the origin of replication. And what that means is that from that origin of replication, DNA replication had to happen towards this replication fork, which we see right here. Okay, so the overall direction of replication starts at this red point, and it started and went all the way like this. Okay, now what we see here is the leading strand at the top, and we'll talk about this one first because it's the easiest one. Okay, and we'll just kind of isolate this picture. We notice that the original parent strand of DNA, this one up here, this dark blue one, we know that it's labeled 3'. Okay? So we look there and say, okay, that's 3'. And what that means is that our new strand, since DNA runs anti-parallel, the new strand, if we're going to run it and match it up, it has to be labeled 5', because that's just the way DNA does. They're anti-parallel. Those edges have to be the opposite of one another. So if this one's 3', this one has to be 5'. And so DNA polymerase, that's this big, chunky kind of peach enzyme right here, will have started right where my red dot was. It started there. And it laid down DNA track, DNA base pairs, starting at the 5' and moving in the 3' direction. Okay, the 3' direction of this new strand. So if my pen thing doesn't work, but if we were in class, I would label this end of the light blue strand right here. I'd put a little 3 with an apostrophe. That would be labeled the 3' end, meaning that if I was going to add another base to this end, it would have to be right here, because you can only add new bases to the growing 3' end. Now, if I wanted to add another one, it would be right here, and then another one right here, and another one right here. And you can kind of see that's what's happened down here at this second picture. Okay. Now, if we look at the other strand, the lagging strand, that's where things get a little bit tricky, because we look at the other template strand down here. It's labeled 5'. Okay, that's natural because this one up here was labeled 3'. We would naturally think that the other side would have to be labeled 5', right? Now, that presents difficulties in creating a long, nice, continuous strand like the leading strand, which we see right here. These strands have to be added in chunks because of how DNA has to be laid down. So if we look at this little Okazaki fragment right here, okay, it's only a little guy, it's only 5 base pairs long. The DNA polymerase would have started right here where my red dot is at. And it would have laid down DNA base pairs in this direction. It would have gone like this, like that, and it would have stopped right there. Okay, because new DNA is laid down in a 5', which is where my red dot is at, to a 3' direction. Okay. Once the DNA polymerase reached this spot right here, it would have fallen off. Okay, it's got nothing left to attach to. It would have fallen off, and then it comes back up here, like almost like a typewriter. And it started a new one right here. It would have started this little chunk right there. It would have started it right here. Okay, and it would have laid down a new base here, and then a new base here, and then a new base here. It's got one more base pair it's going to lay down, which is right here. And the reason it has to make these little chunks like this is because you can only add bases to the growing 3' end. That's what this is. That's what this little red dot is. That's the growing 3' end. So if I want to add another base, it's got to be right here. Okay. Now it's eventually going to run out of room. It's going to run into a chunk that's already been done. And so it doesn't have to stay on any longer. So this guy will fall back off and it'll go back. I call this upstream. It'll go towards the replication fork, and that's exactly what we see here. So this chunk here is this one here. This chunk here is this one right here. Okay? Now at this point it fell off, the DNA polymerase fell off, went back upstream, and started to lay down a new chunk of DNA. And the reason for that is because of this template strand and its chemical nature. The reason that it started out at 5'. Okay? I know that's difficult to understand, but when you see animations, and I'll eventually link animations probably tomorrow, on Tuesday, link animations to help you get a better understanding of what's going on there. I've also got something else we can do if you are confused about the lagging strand, but it makes more sense to see it all together. Now, one thing we have yet to talk about is something known as RNA primers. Okay. Now, these DNAs that we see here created, these light blue segments of DNA, they need a starting point to actually take place. DNA polymerase, which is the pink thing again, or the peach thing again here, needs something to attach to. And so what we need to discuss now is something called RNA primers. Okay? And RNA primers are attached to the template strand by something known as primase. Okay. Now, when we discuss, I'll get all these together. We'll do that now. When we discuss how primase actually works, let's start here. This one, this pink enzyme here is primase, and what it's laying down is an RNA primer. This red thing is the RNA primer. And basically what that is, that's like a starting point. It's like a docking point for DNA polymerase to attach to. Okay, and so that's what we see has happened right here. This RNA primer has already been laid down, and what it did is it allowed this peach-colored DNA polymerase to attach to it. Okay, and what we see in this picture has already happened is that DNA polymerase has already got to work. It's already done some laying down of the DNA track. Okay. Once it runs out of room, and it might, okay, and it does here, it falls off. You can see that it falls off here, and it starts another DNA track until it gets to this little primer. Now, one thing that's not indicated in this picture, and they probably should have put that since this picture does describe RNA primers, is that somewhere up here upstream, there was a little red chunk of RNA that was laid down, just like this little pink enzyme did. That's the only way that this DNA polymerase could have actually attached up here and started to make this little chunk of DNA. Right? And so ultimately, DNA polymerase lays down enough of its track here to meet this little RNA primer, and something else comes in that we're going to discuss here in a second that helps to complete the picture, and then something else here called DNA ligase helps to finish off the DNA strand. Okay. Now, those other things that I talked about in the picture just previously is going to fall under these other proteins that help assist in DNA replication. So I'm going to talk about some of the other things that we haven't yet discussed. The picture does get a little bit more complicated. There are other enzymes and proteins that do help with this process, and they all have a very specific little job. One of the first ones we have yet to talk about, and probably should have done so when we discussed the origin of replication was an enzyme known as DNA helicase or helicase. And what helicase does is it untwists the double helix, okay, so it flattens it out, and it also unzips it, separates it, as you can see by this totally kind of inappropriate joke here. We'll just move on. Let's see. Okay, a couple of other ones that are important here, known as single-stranded binding proteins. Um, and single-stranded binding proteins, topoisomerase and primase. Primase we've already talked about. Topoisomerase and single-stranded binding proteins are also two important ones that we need to discuss a little bit what they do. Now, when helicase after helicase unzips the DNA strand, the thing wants to naturally just bond back together. It's kind of what it does. It has these forces of attractions, just like magnets. You pull magnets apart, they want to come back together. You pull static electricity, stuff that has static electricity apart, it likes to come right back together. Single-stranded binding proteins cause an effect that basically prevents that, keeps it single-stranded for a little bit of time. Topoisomerase, I wish we were in class. We could get you guys up out of your seats and active, and maybe I'll have my kids do this live. It would be fun to do. But topoisomerase is something that corrects overwinding. If you've ever twisted up some cord, like if you've ever taken like electrical cord and twisted it up, I know this happens to me a lot. What happens is if you twist it up too much, it balls up on each other and it makes big knots. And what topoisomerase does is it corrects this, because that's exactly what's going to happen when you unzip the DNA right down the middle of something that's already twisted. It's going to super coil it. It's going to overwind it further downstream. And what topoisomerase does, it corrects that overwinding issue. And that overwinding issue can cause knots, it can cause breakages, all sorts of stuff that's bad to the DNA. And again, if we were in class, you could see that demonstrator right in front of you, and it would make a lot more sense. And this one here, primase, we have already talked about. Now, DNA polymerase 3, that's what this stands for, DNA pol 3. DNA polymerase 3 is the one that I've been talking about up until now. DNA polymerase 3 is the specific name of it, okay? So that's nothing different than what we've already discussed in terms of DNA polymerase. Basically, what it's designed to do is synthesize leading and lagging strands of DNA. That's its main job. Now, there is a second type of DNA polymerase called DNA polymerase 1. DNA polymerase 1 has a very specific job. Those little RNA primers that I talked about here that were red, that were laid down by the primase, those things can't be there by the time this is all said and done. Okay? They're not DNA, they're RNA, so they have to be replaced. And that's what we see down here, this little yellow guy. This yellow one is called DNA polymerase 1. And what DNA polymerase 1 does is it takes these little RNA primers right here, these little RNA base pairs, and it replaces them with actual DNA. Okay, so it fixes that problem. DNA polymerase 3 needs a place to dock, and DNA polymerase 1 removes that docking space and puts actual DNA there. Okay? And then this other one we see right here, DNA ligase. That's the one I passed up down here as well. It's hard to see with this guy in the way, but DNA ligase is that kind of bluish one, and DNA ligase is going to seal, and we can kind of see there's little gaps here, is going to seal the gaps in the DNA backbone between the Okazaki fragments and other Okazaki fragments or the leading strands. Okay, so DNA ligase is like the glue that will actually glue the little chunks of DNA together that were there because of the way that DNA has to lay down base pairs. Okay, so here we can see all this stuff together. We can see everything. I like this picture as something that you can study from. It shows you the overall direction of replication. It labels both ends of the DNA, shows you the leading strand, what's happening with DNA polymerase 1 over here. These black ones here, I believe, are single-stranded binding proteins. I think this green one right here is helicase. It's unwinding and unzipping. This is stabilizing the single-stranded conformation. We see here primase. Primase is laying down an RNA primer. Okay. This DNA polymerase 3 is laying down some DNA track. When it gets to this primer right here. It's going to stop. It's going to fall off. And it's going to go back upstream and look for this one. And then it's going to lay down this chunk. And when it gets here, it's going to go off and lay down this chunk. Okay, so it does this side in chunks. We see DNA polymerase 1 right here. It's removing those RNA bases. They were right here. We can see that now those red ones have been changed to light blue. And we see DNA ligase come in and seal the gap right here. DNA ligase will glue that gap in the backbone together. So that's how that process happens. A lot going on, a lot of moving parts. Again, I'm not going to do the animations here because it slogs down the upload. So it really is kind of amazing when you think about all of the base pairs that we have. I think it's like 3 or 4 billion base pairs, something crazy like that. And for the most part, our mechanisms of DNA replication get it right almost all the time. It's very rare do we actually have DNA mutations that cause us any harm. But even if we do, there are ways that our DNA can fix it. Okay, the DNA polymerases, both 3 and 1, will proofread that newly made DNA, and it replaces anything that's incorrect with something that is correct. Okay, and so in something known as mismatch repair, um, these enzymes will actually go back and proofread and correct the base pairing. If there's any errors, it'll correct them, and it does so by a process known as nucleotide excision. Okay, and nucleotide excision uses another type of enzyme called nuclease, and what nuclease does is like a little pair of DNA scissors, and it'll cut out these ruined parts of DNA. So right here we see kind of a mistake. Okay? These two base pairs somehow got glued together and they didn't bond properly to the other side of the template. Our DNA polymerases are going to proofread that and say, okay, something's wrong here, and they're going to set in motion the way to fix it. Okay, they're going to get in here nuclease. That's this kind of greenish minty colored one, and nuclease is going to chop this little chunk out. Right there, you see, it's going to get rid of it, and DNA polymerase is going to come in. And my wife's giving me a hard time right now, giving me stupid looks. I know I'm a talker, okay, and you just made it longer. But anyway, DNA polymerase comes in and adds these new bases right here, and then we have DNA ligase come in and seal the whole thing up. So DNA polymerase, and you can see by the colors, DNA polymerase 1 comes in and adds those bases. So we do have ways of fixing mistakes when we see them, and the DNA polymerase, again, is instrumental in that as well. All right, again, I'm going to try to collect a bunch of animations for you guys to watch. But as for right now, I'm going to let this thing upload, and hopefully there's no issues with that.